Good evening students. So in this video we are going to discuss how antidiuretic hormone acts on collecting duct to reabsorb water and how antidiuretic hormone helps in keeping the urea cycle running. For understanding the mechanism of ADH, we need to understand various steps. Let's discuss them one by one, right? Let's first of all draw our cell, right? We will draw a big cell because we have to write many things, right? Okay. Now, which hormone is acting? ADH, right? Antidiuretic hormone is acting on a cell of collecting duct, right? Now, what's the name of our receptor? It acts on V2 receptor, right? What's the name of our receptor? V2 receptor. Right? And what kind of receptor is V2 receptor? It's a G protein coupled receptor, GPCR, right? Now what this V2 receptor is going to do is that it's going to increase the level of adenyl cyclase, right? Adenyl cyclase, right? What adenyl cyclase is going to do? The adenyl cyclase will convert your ATP into cyclic AMP. Right? What cyclic AMP is going to do? The cyclic AMP is going to increase the level of protein kinase A. Right? It will increase the level of protein kinase A. Right? What protein kinase A is going to do? The protein kinase A will cause phosphorylation of proteins, right? Phosphorylation of proteins. Right? And what phosphorylation of proteins is going to do is that you will have a lot of vesicles, right? You will have a lot of vesicles over here. These vesicles will be having aquaporin 2 channels, right? These vesicles will have aquaporin 2 channels. So it will cause the recruitment of these vesicles towards the membrane. So phosphorylation of proteins will cause recruitment of the vesicles containing aquaporin 2 channels towards the membrane, and the aquaporin 2 channel will get inserted into the membrane. And what will happen after that? The water will get reabsorbed from it. This is the luminal side, right? On this side you have got urine, the filtrate which will ultimately become urine, right? So the water will get reabsorbed through the aquaporin 2 channel, short form AQP2, right? Now once the water reaches over here, right, it will, this is the interstitial side. On this side you have got interstitial, right? And apart from that, the vasa recta, blood vessels, right? So on this side you will have two water channels, aquaporin 3, AQP3 and AQP4. Right? Two water channels are there, AQP3 and AQP4. So water will come inside through aquaporin 2 channels. It will travel and it will exit the cell into interstitium with the help of aquaporin 3 and aquaporin 4 channels. From here your water will enter into the blood vessels with the help of osmosis and bulk flow into the vasa recta and go inside the blood, right? Apart from that, this protein kinase A, this cyclic AMP to be in particular, this cyclic AMP will act on nucleus, right? And it will increase the formation of aquaporin 2 channels, right? Formation of AQP2. So ADH has got both short-term effect and long-term effect. In short-term effect, it increases the recruitment of aquaporin 2 channels. In long-term effect, it acts on nucleus and increases the formation of aquaporin 2 channels. So, how exactly ADH is acting? ADH is acting on your V2 receptor, which is a G protein coupled receptor. It is increasing the level of adenyl cyclase. This adenyl cyclase in turn is converting ATP to cyclic AMP. This cyclic AMP is increasing the protein kinase A, which is causing phosphorylation of proteins, right? And this phosphorylation of proteins leads to recruitment of the aquaporin 2 channels to the cell membrane, and water enters through these channels and exits the cells through aquaporin 3 and aquaporin 4 and ultimately enters the blood vessels. Cyclic AMP also acts on nucleus and increases the formation of aquaporin 2 channels. But how exactly ADH is helping in running of urea cycle, right? This phosphorylation of proteins also leads to recruitment. You have got vesicles which have got another ion channel called UTA1, right? So it will, the urea over here, right? Urea over here will come through UTA1 and exit the cell through UTA3. Right? It will come inside through UTA1 and exit the cell into interstitium, right? Through UTA3. Right? Now, this is a luminal side. That means from the urine side, the urea is getting reabsorbed and it is exiting the cell through UTA3 going into interstitium. From interstitium, it is going to enter into your thick ascending limb of loop of Henle, right? And from once it enters your thin ascending limb of loop of Henle, it is going to enter thick ascending limb, DCT, collecting duct, and will come back over here. Then again UTA1 going inside the cell, exiting through UTA3, interstitium, then again your thin ascending limb of loop of Henle, right? To make the things clear, I'll just draw a small diagram of nephron over here, right? Small diagram of nephron. If I draw, it will be a lot more clear, right? PCT, then your descending limb of loop of Henle, ascending limb of loop of Henle, right? And collecting duct, right? So this urea will actually get reabsorbed from the lumen with the help of these two transporters, UTA1 and UTA3, right? And then they will enter the thin ascending limb of loop of Henle, right? And then thick ascending limb of loop of Henle, then into DCT, and then into the collecting duct. So this is a urea cycle, from collecting duct into interstitium, from interstitium to thin ascending limb of loop of Henle, thick ascending limb of loop of Henle. It circulates like this. And now why we need urea to circulate like this? Because in peak ADH action, this urea constitutes roughly about 40 to 50% of the osmolarity of this interstitium. So it helps in reabsorption of water, right? So it helps in concentration of urine, right? So if you have lot of urea in this medullary interstitium, the kidney will find it easy to concentrate the urine, right? So ADH helps both in water reabsorption and also in urea the recycling of urea, right? So let's sum it up, right? Your ADH, the ADH will act on V2 receptor. V2 receptor will increase your adenyl cyclase. Adenyl cyclase will convert your ATP to cyclic AMP. This cyclic AMP will increase the level of protein kinase A. This protein kinase A will cause phosphorylation of proteins. This phosphorylation of proteins will cause increased recruitment of the aquaporin 2 channels, right? On the luminal side, the water will get reabsorbed through these aquaporin 2 channels and will exit the cell into interstitium with the help of aquaporin 3 and aquaporin 4. This water will ultimately reach vasa recta through bulk flow and osmosis, right? The cyclic AMP will also act on nucleus, right, and cause increased formation of aquaporin 2 channels, right? The ADH also causes recruitment by the help of this phosphorylation of proteins. Protein kinase A will cause phosphorylation of proteins, and this phosphorylation of proteins will cause increased recruitment of UTA1 receptors. This will absorb urea from the lumen. It will exit the cells through UTA3, right? And it will ultimately reach your thin ascending limb of loop of Henle, thick ascending limb of loop of Henle, DCT, and collecting duct, and the cycle will repeat itself, right? So hopefully this video has helped you in understanding. One very important point I want to discuss over here is, suppose this V2 receptor gets defective, right? Suppose if this V2 receptor gets defective, the ADH won't be able to act on the V2 receptor, right? So in that case, what we will say, this is a case of nephrogenic diabetes insipidus, right? Nephrogenic diabetes insipidus. I'll write DI, right? If ADH is deficient, we will call it central diabetes insipidus. But if ADH is not able to act on V2 receptors due to defective V2 receptors, we will call it nephrogenic diabetes insipidus. So this much in brief. Hopefully this video has helped you in understanding the mechanism of action of ADH and its role in reabsorption of water and its role in urea cycle and central diabetes insipidus and peripheral diabetes insipidus. Okay, wish you all the very best. Thank you.